All right, so for the next week or so, cell transport is our topic. How do things move through a cell? And what we talked about yesterday, we'll review a little bit today. Before you can understand how things move in a cell, you've got to understand the cell membrane, because it's like the gateway into the cell. We've used over and over again this word when we talked about the cell membrane. Actually, a phrase, selectively permeable. Some things can get in, some things can't. Some things can get out, some things can't. And we talked yesterday about some factors, some things about a chemical that determine whether or not they can get in or out. And these were the two main ones we talked about yesterday. In terms of size, what kinds of things can go in and out with no problems? Small. Really, really tiny things. So smaller things can go through the cell membrane pretty easily. Large things, though, and there are a lot of large molecules that your body needs, they can't. They can't go through the main part of the membrane at least.
So it's like our cells are eukaryotic and like prokaryotic smaller than eukaryotic, so they can get in faster, like bacteria and stuff.
And we're not talking about cell. We're not talking about a cell getting into a cell right now. We're talking about a chemical getting into a cell. So we're saying small chemicals can go into cells. Big ones have a harder time. We're not talking about one cell getting into another cell. All right. The second thing we looked at was polarity. If something is polar, what does that mean about it? It means it's charged. It's got a positive side and a negative side. Well, most of the cell membrane isn't polar. Most of the cell membrane is non-polar. So what kinds of things can go through easy? Charged things or not charged things? Non-polar things, not charged. So non-charged things, non-polar things, they can get through pretty easy. Things that are polar or ions, it's hard for them to get through. We'll talk about some of this receptor stuff later. All right, the basic structure of your membrane. It's made of a bilayer of what things? What are those things? What kind of oil? What kind of lipids? Yeah, what do they call them? What kind of lipids? Phospholipids. Phospholipids. Phospholipid bilayer. And it's sort of like vegetable oil. It's not hard like a cell wall. Cell membranes aren't hard like a cell wall. Kind of fluid. This is trying to show you what kinds of things can get through that phospholipid bilayer. Here's a molecule that's not charged. Notice it goes straight through. It can go in, it can come out. No problems. Here's a molecule like water. Even though it's polar, it's really wet. Water's polar, but it's also very, very wet.
Basic.
Very small. It's only 3 atoms big. So small things, they tend to be able to go in and out pretty easily. But these big things, like this is a protein right here, for example. It's really big and bulky. It can't go in and out, at least not through the phospholipid bilayer. And these little things over here are ions, like maybe Na+, Ka+, Cl-. They've got a charge. Charged things can't get in and out very easily at all. Now, are there big molecules that need to get into some cells? Yeah. Are there charged molecules that need to get into some cells? Yes. So there's another way in, besides just going through the phospholipid part of the membrane. And when we talked about membranes earlier, we'll get deeper into this later. Let's go to a picture before I try to describe this. In this picture, what are all the blue things inside this picture? They're proteins. And some of those proteins, for example, maybe like these right here, sometimes these are called channels. Channel proteins. And now you know what kinds of things probably go through those channels. What kinds of things can go in and out of the cell through the channel? Things that are charged or things that are big. That's how they get in and out. Think of it like a tunnel for those kinds of things. Turns out there's a separate tunnel, a separate channel for each different thing that needs to get in and out of the cell. They're specific. For example, a channel that would let chloride ions in, it won't let anything else in. It only lets Cl minuses go through. A channel for glucose will only let glucose go through, pretty much. They're specific. The channels are very specific. All right, so let's get back here for a second. So we also yesterday talked a little bit about diffusion, and we said this about diffusion. We said it's the movement of substances from high concentration of that substance to low concentration of that substance. We talked about a concentration gradient. What did we say the word gradient means? Talking about like the grade of a road. What did we say that means? So the difference in elevation. Gradient means a difference in something. In this case, we're talking about a difference in what? It's a concentration gradient. So we're talking about a difference in concentration. When we're talking about diffusion, another way of saying it is that diffusion is the movement of a substance down a concentration gradient. That's just another word, another way of saying it's moving from high to low. Sometimes instead of saying down a concentration gradient, some books will say that it moves with the concentration gradient, going from high to low. All right. We also talked about some places where diffusion is important in your body. For example, we looked at your lungs yesterday. And we call those little air sacs that fill up your lungs alveoli. We said those little air sacs touch and come in contact with what? Little bitty blood vessels. Capillaries, which we see down here. When you breathe in air, most of what you breathe in is nitrogen, right? But what's the second most abundant gas? Oxygen. There's a lot of oxygen in the air you're breathing. It's like 16% or so is oxygen. So there's a lot of oxygen in these little sacs after you take a breath in. So we'll say high O2, high oxygen in those sacs. But where's the blood coming from that comes to the lungs through those little capillaries?
Are those red things capillaries as well along the blue?
These little rings?
Yeah.
Now, anybody know what those little rings are? We'll do this for me. Take a second and kind of run your hands up your throat. The tube. Run your hands up your trachea.
Your esophagus?
You feel the little rings on your trachea? You should feel some little rings, little bumps on there. Why would you have little rings on your trachea? Yeah, they're little rings of cartilage. So that if somebody came up and popped you one in the throat, what do you hope those rings do? You hope they keep your trachea from doing that, okay, from collapsing. Because if your trachea was to collapse, you're not getting any oxygen to your lungs, and you're going to die pretty quickly. Instead of support. What now? What about it? Yep, that would be bad too. But the point being, those are little cartilage rings, partly. That some of it may be supposed to be blood vessels too, but yeah, go ahead.
In and out of that, doesn't the inside swell?
Yes, and the hole inside closes, or almost closes. All right, so back to what I'm saying though. Blood coming to your lungs in the capillaries. Where's that blood coming from? Yeah, it's already been around the blood. It's already been used blood, right? Which means what's very low inside those blood vessels? Low oxygen. But because it's already been used, what's really high inside those blood vessels? Carbon dioxide, right? CO2. So there's low oxygen inside these capillaries, high CO2. How much CO2 is in the air you're breathing right now? Not much. So that means that in your little air sacs, there's low CO2. So in terms of the way the gases are going to move, there's a lot of oxygen in your lungs, not so much in your capillaries. So the oxygen moves into the capillaries from high to low. On the other hand, there's a lot of CO2 in the capillaries and not much where? In the lungs. So the CO2 goes the other way, from high to low. And that's how your body exchanges gases. Simple process. It's just simple diffusion. Same kind of thing happens to some extent. Where does your nutrients that you eat, where do they get into your blood? What organ?
The intestine.
Which intestine?
Small intestine.
Smaller. So it's a little more complicated than this, but this is an oversimplification. Your small intestine, the walls of it look like this. It looks almost like little hairs and little teeth inside. It's very tiny though. And inside those little hair-like structures are capillaries. So let's draw those in red. So these little squiggly things I'm putting inside are supposed to be blood vessels, capillaries. So as the food that you've eaten moves through the small intestine, it's getting digested. Maybe digestion is almost through. See how it's got to kind of like climb over these little things to get through your intestine? As it's climbing over them, what's happening to a lot of the stuff that's in that gooey, what's left of your food? It's being moved from the intestine into those capillaries. And one of the ways that happens is by diffusion. Because there's going to be a lot of nutrients here, not so many nutrients in these little blood vessels. And how do we know they're going to move?
High to low.
From high to low. From the intestine into the bloodstream. So diffusion's important all over your body. It's the way we exchange gases. It's the way we absorb our nutrients. It's the way we move waste out of our blood and into like our urine and such. So diffusion is hugely important in your body. I'm going to take a second and pause the video. All right, the next thing that we need to talk about is something called osmosis. Osmosis is a special kind of diffusion. It's just the diffusion of good old water. Osmosis is simply the diffusion of water, which means water. Notice it says here, diffusion of water through a selectively permeable membrane. If it's a form of diffusion, water has to be moving which way? From high. We got to be more exact when we're talking about osmosis, though. It moves from high water concentration to low water concentration. But sometimes when we're thinking about osmosis, we don't always talk about it in terms of the water. We talk about it sometimes in terms of what's in the water. What do we call things that are dissolved in water?
Solvents.
Solutes, right? So sometimes we think about it in terms of the solutes, the things that are dissolved. For example, let me put in a blank slide here and draw a picture. Let's think about the bag that we just had for a second. So here's what we started with. We started with a beaker. And we have water in the beaker, just regular old water. And we put into that beaker that dialysis bag. Now remember, what did we have in that bag? Sugar and water, right? So there's some H2O in here. And there's also sucrose. Now under normal conditions, what should happen to that sucrose? Sucrose wants to come out, right? Because there's high concentration in the bag and there's zero concentration in the beaker. But I'm telling you, I know because I've done this a bunch of times, sucrose is too big. It won't fit through the little pores in the bag, so it's stuck. Is nature like that when concentrations are not equal? No. So if the sucrose can't move, what's nature going to do to try to even out the concentrations on each side of that bag? Can't make the sucrose move, so it makes water move. Now think about this for a second. There's a higher concentration of water out here, right? Because it's just water. out here in the beaker. There's other things in here besides just water. So water moves from high concentration of water to low concentration of water. As the water moves in, let's just do a demo right here real fast. Let's take a little bit of water and a whole lot of sugar. As you can obviously see right now, the concentration of sugar in this is very high, right? Let's say this is what's in my bag to begin with. This is what's in my little bag, my dialysis bag. But we said over time, since the sugar can't move, water starts to move in. So let's move a little water into this. Stir it up. As I put more water into here, what happened to the concentration of sugar in here? It got lower. And it's getting closer and closer to the sugar solution outside the bag, right? So that's nature's way of trying to even it out. Even though it can't move sugar, it can move water. And what do we call when we keep adding water to something? What's the sciencey sounding word for it? When you make something less and less strong because you keep adding water to it? Diluting it, right? So this water is diluting the sucrose concentration. If we add a little more water to this, it becomes more and more dilute. And the difference in those two concentrations becomes less and less, right? Now, in reality, is this ever going to get to equal concentrations? It can't, right? Because the sucrose can't move. But water is going to continue to move in, continue to move in, continue to move in, and it's going to dilute that sucrose and dilute that sucrose and dilute that sucrose as far as it can. Now, if this was one of your cells and it had sucrose in it like that, and the sucrose couldn't get out, what's eventually going to happen to that cell? It's going to burst, right? It's just going to keep getting water, keep getting water, keep getting water until it explodes. Now, on the other hand, if it's a plant cell, it's not going to quite look like this, right? It's going to have what around the outside? It's going to have a wall around it. Now, things can go in and out through the wall, no problems. But what's the wall going to keep from happening? It's going to keep the cell from exploding. It's going to let that cell swell up and swell up and swell up until eventually it bumps up against the wall, the inside of the wall, and the wall protects it from bursting. That's one of the main functions of cell walls and lots of things, is to keep them from bursting because they're absorbing too much what? Water. That's important. Other than plants, what other kinds of cells have walls? Bacteria and some protists, yeah, and fungi. So it turns out that most kinds of organisms have cell walls. Animal cells don't. We're about the only type of cell that doesn't have the wall. Most other things do. So walls are pretty common. But let's talk about another way of defining osmosis. If we define it in terms of the water, it's the movement of water from high concentration of water to low concentration of water. That makes sense, right? But sometimes we define it in terms of the solute. So out here, in the beaker, out here, what kind of solute concentration is there out here? To begin with, what kind of solute concentration out here in the beaker? 0. So we're going to say out here is low solute concentration, but also high concentration of what? Water concentration. In this bag is a low concentration of what? Water. But in that bag also is a high concentration of what? Sucrose. So we're going to define osmosis two ways. It's the movement of water from high water concentration to low water concentration. So the water is going to go into the bag. But another way of thinking about it, it's the movement of water from where there's a lower solute concentration to where there's a higher solute concentration. If you have a solution that has a low concentration of solutes, what's the solution mostly made out of? It's got a low concentration of solutes. What's the solution mostly made out of?
Water.
It's mostly water. If on the other hand it's got a high concentration of solutes, there's not very much what there. There's not very much water. There's just two ways of saying the same thing. Don't get confused by that. Osmosis is the diffusion of water from high concentration of water to low concentration of water. Or, another way of saying the same exact thing, it's the movement of water from where there's low concentration of solutes to where there's high concentration of solutes. Remember, what are solutes again? What's a solute? Yeah, it's just like sugar, salt. It's something that dissolves in the water. So if there's less things dissolved in the water, that means there's a lot of water there. If there's a lot of things dissolved in the water, that means there's not as much water, right? So two different ways of saying the same thing. Let's go back and get a couple of words here before we take a time out. Let's say that I talked about, kind of pick on here. Let's say that I talked about Joyce, and I said Joyce is hyperactive. That'd be kind of true, right? What does hyperactive mean?
Like hyper,
Just what does hyper mean?
very fast and energetic.
Hyper means more than normal, okay? She's more active than normal, okay? So we're going to talk about a word here called hypertonic. Hyper means more than. And tonic refers to the concentration of what? Not water.
What?
The concentration of solutes. So if we say a solution is hypertonic, it's got more solutes than whatever we're comparing it to. A solution is hypertonic if it has more stuff dissolved in it than whatever we're comparing it to does. Does that make sense? Hypertonic. More solute concentration than what we're comparing it to. Let's say that I said that that James had hypoglycemia. Hypoglycemia. What do you think that means, Ryan?
Too much sugar.
Not hyper. Hypo. It means he has low blood sugar. Hypo. Hypo means less than normal. So if we call a solution hypotonic, following, we have a solution that's hypotonic, that means it has less solutes in it than whatever we're comparing it to does. So this means less solutes than what we're comparing it to. Hyper more, hypo less. What's ISO mean? ISO means equal. For example, Aaliyah a minute ago had to go get some contact solution put in her eyes. Contact solution is, what's another name for it? Saline solution, which means salty water, right? I should have just came back to the room. Aaliyah needed some saline. I should have just came back to the room and go back in the back and get some salt, pour it in the water, and say, here you go, Aaliyah, right? It's saline solution, it's salt water, what's the difference? Alright, the saline solution that you buy at the store, in the bottles for contacts, is specifically made to be isotonic to your body fluids, which means it's got the same amount of what in it? Same amount of salt as your body fluids, same amount of solutes. If you pour saline on your eye, what do you feel? Nothing. It gets wet. Your eyeball gets wet. It doesn't burn. It may, it's kind of irritating when you drop anything in your eye, right? But
Yeah.
it doesn't really burn. It's the same concentration as your eye. It's made to be that way. On the other hand, if you put really salty water on your eye, what's going to happen? It's going to burn like a devil, right? Let's think about this for a second. Let's say that you've been exercising all day, and you're really sore. Maybe you've been lifting weights. So you go home, you get one of those big jacuzzi bathtubs. You hop in, you turn on the jets, you kick back, You take a nap in the tub. That's not a good idea. Don't really do that. But you've been soaking in there for like 30 minutes in the jacuzzi, and you get out. And when you get out, you look at the pads on your fingers and your toes, and they all look wrinkly. And so thinking about what I just talked about, think about what I want you to take just a minute and think about what I just mentioned. Think about why that happens. No, don't talk about it. Just think about it for a second. You've been in a tub for 30 minutes. You get out and your fingers and toes are all wrinkly. I want you to come up with a response for me for what's happening. And I'm going to call on somebody here in just a second to tell me what you think. And I'm going to tell you that what you think is probably not what's really happening. But think about it first. Think it through. Why do the pads of your fingers and toes get all wrinkly when you're soaking in the tub? I want everybody to think about it for just a second. I'll let you answer, and I'll let Josh have a shot. All right, so let's do this. All right, Mr. Childers, take a shot at it.
I looked at someone it's true facts.
Okay.
So this true facts, and it said that it's not like that water makes your hands all wrinkly. It's that your body naturally makes your hands that way, so you can have more grip underwater.
Okay, but there's still something's got to be happening with the water, though, right? Josh, what do you think?
Maybe I think that.
Let's listen to Josh, guys.
I think that whatever you're bathing and swimming in, that if you stay in it long enough, the water will soak into your skin and take out the natural oils.
Okay. But that's probably true. But there's a little more to it than that. Most people, when I ask this question, they tell me what's happening to your fingers. What would your average person say? They say shrivels, right? Exactly. Most people say shrivels. Think about this for a minute. Here's my beautiful rendering of your hand in the bathtub. Okay? Out here is just good old water, right? Good old water. So you're just taking a regular bath. There's not hardly anything in there. Hold up. There's not anything in the bathtub except maybe a few little salts and some soap. It's water. What's inside your hand? All kinds of stuff. Sugars, proteins, salts. Lots of what inside your fingers? Lots of solids. How does water move? From where there's less solutes to where there's more solutes. From where there's a higher concentration of water to where there's a lower concentration of water inside your fingers. I'm going to tell you, your fingers aren't shriveling. Your fingers are swelling. Your fingers are not shriveling up when you're in the bathtub. They're swelling up. Here's the thing. Most of your skin, listen, most of your skin has this stuff in it that makes it waterproof. That effect doesn't happen to the front of your hand, right? It just happens to the pads of your fingers.
Actually, does if you stay in it long enough.
Not to the same extent. Most of your skin has this stuff in it called keratin. It's a protein that essentially makes your skin what? Waterproof. But keratin is kind of hard. It's the same stuff that's in your fingernails. Would you want to put that on the pads of your fingers? Why not? You couldn't feel the thing. I mean, the pads of your fingers are one of the most sensitive parts of your body, right? You need to be able to feel things. You need to be able to grip things, like you were saying. They don't have that keratin in it, at least not nearly as much. Which means the pads of your fingers are not waterproof. Which means, but there's some strips of keratin in there. So water starts to move from the tub where there's a whole lot of what? A whole lot of water, a high concentration of water, toward the inside of your finger where there's not as much water, right? Water moves from where there's not hardly any solutes in the bath water to where there's lots of solutes inside your finger, and your fingers start to swell. It looks like they shrivel because there's some strips of keratin in there, and the parts that don't have the keratin do what? They swell, but the parts that do have the keratin, they don't. So it's not shriveling, it's swelling. Say again? No, it's just where water has moved in and sort of blown up your fingers like a balloon. So you're not shriveling when you're in the bathtub, you are swelling when you're in the bathtub. Water moves in to parts of your body that don't have keratin, that's not waterproof. Water always moves from high concentration of water to low concentration of water. It always moves from where there's less solute concentration to where there's more solute concentration. And let's throw another definition here at it too. Another way to define osmosis is that right there that I just circled. Water moves from hypotonic solutions to hypertonic solutions. That's saying the same thing, right? What do we say hypo means? Hypo. Less, in this case, less what? Less solutes. To hyper, which means? More solutes. So all three of my definitions, whether we're talking about high concentration of water to low concentration of water, low solute concentration to high solute, or hypotonic to hypertonic, they all are saying the same thing. Just three different ways of saying the same thing. Think about one more example. We sort of talked about this a little bit yesterday. Let's say you're living back in the 1500s. You just killed a deer. It's fall. You killed this huge deer. There's no way you and your family can eat it all right now. You don't know when the next deer comes along. It may be 2 months before you find another deer to kill with your spear. So you don't have a refrigerator. There's not a lot of ice around. You don't want that deer to spoil. So what you do is let's say this is a big hunk of deer meat right here. Isn't that a beautiful piece of artwork?
Oh yeah.
Okay. Beautiful hunk of venison. And what you might do, you gotta be creative and thinking here. I'm working on you developing an imagination.
Do you want to go to art with me next class?
No. I wouldn't want to intimidate Mr. Cofer with my skills.
Oh.
Sarcasm. Regardless, what do you do with this big hunk of venison to keep it from spoiling? Put it down in a solution of really, really salty water, or maybe really, really what? Sugary water. Something with lots of solutes in it. And probably you may not even put it in, you may just pour it all over it, right? What happens to the water that's in that meat?
It goes out.
Gets pulled out, right? From the water that's in the meat gets pulled from where there's less solute inside to where there's more solute on the outside where all the salt's at, right? So that it dehydrates it. Makes jerky out of it, essentially. Well, that by itself doesn't mean a whole lot. What spoils your food?
Bacteria.
Germs, okay? Fungus, bacteria, things that try to eat your food before you do. We said yesterday, if this is now really salty or really sugary, and a little bacteria lands on this piece of food, what happens when that little bacteria lands there?
The salt goes and gets it.
The water that's in the bacteria gets pulled out toward the salt because water moves from where there's less solutes to where there's more solutes. From where there's a higher concentration of water to where there's a lower concentration of water. And essentially, when the water gets pulled out of it, what happens to the bacteria? It dies, and therefore you don't have to worry about it eating your food because you just killed it, right? You killed it with the sugar. Okay, so that's a way to keep spoilage from happening is either making things really salty, really sugary. Another thing we'll mention is in plants. Actually, let's talk about this. Let's talk about fish for a minute. Talk about fish. I'll amaze you with another beautiful artwork here. Let me try that again. That's terrible. Let's see. Beautiful fish, right? All right. Anyway, hold the thought. This is a freshwater fish. So out here is freshwater. It's a brim.
Maybe a bass.
Okay, it's a bass. Sure. It's a smallmouth bass. All right. Anyway, this thing's in freshwater, which means outside its body is pretty much just water, but inside its body is what? Some water, but also some salts and some sugars and some proteins and all kinds of solutes, right? So if you're a freshwater fish, what's the water in the pond wanting to do to you? Is it wanting to come out or go in? Remember, water moves from higher concentration of water to lower concentration of water. From where there's less solutes to where there's more solute concentration. So the water goes in. So a freshwater fish has a constant problem of water always going in. We better have a way to deal with that, right? Because if it doesn't, what's going to happen? It's going to swell up and die. How does the freshwater fish deal with that? The scales help slow it down, but they don't stop it. Freshwater fish urinate all the time. Okay? They are constantly urinating. Okay? When you go swim in a freshwater pond, you're swimming in fish urine. Okay?
Reminder teachers, students are to remain in first block until you're told otherwise.
Okay, so we're saying so. Anyway, back to what I was saying. Freshwater fish, the only way they can keep from swelling up and blowing up is to urinate as much as possible. Keep that water out. Oh, that's where I'm going. Now let's flip it around. Now you got another issue. This time we're going to deal with a saltwater fish. There you go. So now we have not a freshwater bass, but we have a sea bass.
Yes.
And it's living in salt water, which means there's a higher concentration of what out here? Higher concentration of salt out here, a lower concentration of salt in here. All right, well, if there's a lower concentration of salt, there's a higher concentration of what? Water. So we'll say this is high H2O concentration. And out here is low H2O concentration. So if you're a saltwater fish, what problem do you have to deal with? What's water trying to do to you? It's always trying to go out. So saltwater fish, the water is constantly going out of their body. And they're gonna have problems dehydrating. Sort of like the venison soaked in the salt, right? It's the same kind of problem. So how does a saltwater fish make it through that?
Never urinate?
He almost never urinates, and he's constantly what? Drinking.
So it's better to go to a salt.
No, we're not saying that, and I'll get to that in a second. But a saltwater fish is constantly drinking and almost never urinating, so it doesn't dehydrate. Now, a really mean thing to do would be to take a saltwater fish and put it in freshwater, or a freshwater fish and put it in saltwater, right? Because they're going to cause some serious issues for both. There are a few that somehow switch this up, right? For example, the bull shark. Bull sharks, there have been shark attacks near St. Louis. How far is St. Louis from the ocean? I don't know, but hundreds of miles from the ocean. Okay? There have been bull shark attacks on the Mississippi River near St. Louis. way, way, way away from salt water.
I don't know how they get that far. They have a special organ in their body that allows them to soak up salt water and release it whenever they need to.
And there's a few kinds of fish that can switch gears from being salt to fresh, but most can't. Most have to be in one place or the other because it's a big problem. Yeah.
Saltwater crocodile there, is it just salt?
I think the saltwater crocodiles are just saltwater. Don't quote me on that. I'm not sure. I'm guessing.
Well, my brother used to work at this fish store, and then he had this one guy who turned a freshwater catfish into saltwater.
Well, catfish in general are a little more tolerant of that. I was down in Florida this year, fishing a lot down in Florida this summer. And if you fish in like a bay, where you got a river coming into the ocean, you catch a lot of catfish there. They're more tolerant of salt than a lot of fish are. Most fish are not. Most fish, if they hit the ocean and they're a freshwater fish, it's over for them. And the same for a saltwater fish in the river. It's over for them pretty fast. A few things can switch back and forth, not most. Let's go a little bit further here. Let's talk about plants. Actually, let's talk about little protists. Let's say this is a little amoeba. An amoeba is a little one-celled organism, right? One eukaryotic cell. Where do amoebas live? What kind of water? They live in fresh water. There's been some stories the last few weeks that might interest you about amoebas. There's this one kind of amoeba. It's called the brain-eating amoeba that you can catch from swimming in fresh water, like a lake or a pond, and it goes in through your nose or through your mouth, and eventually can get into your bloodstream, and it gets into your brain, and it essentially starts destroying your brain, and you die. There's no good treatment, no good cure. Scary stuff. What else?
Oh, what about that story of the girl that got something like that, and they started eating her eye, and there's another one.
I know about the flesh. There's a kind of bacteria that tends to eat flesh. I don't know about the eye. I hadn't heard of that one.
Yeah.
I don't know.
She was supposed to go to like some Jonas Brothers concert.
Yeah.
But her eyes changed like so bad that she couldn't even see. So it got her ears straight, and she had some bacteria. So it was in her eye.
It was probably heavenly punishment for liking the Jonas Brothers. Anyway, I'll get some more questions in a minute. I'm just joking. Let me finish real quick. These amoebas live in fresh water. An amoeba like you has what inside it? Water, but also some what? Some solids like sugar and salt and proteins and stuff. So in here are some solutes. It's living in fresh water where there are almost no solutes. So what's constantly happening to this little amoeba? What's water doing? Going in or coming out? Water moves from where there's less solute concentration to where there's more solute concentration, all right? Water's going in. And if this little one-cell amoeba doesn't watch it, what's going to happen? He's going to blow up and explode. The cool thing about a lot of little protists and also some plant cells is they have these little organelles in them called contractile vacuoles. What do vacuoles usually do?
Store water.
Store stuff. Okay? These vacuoles store stuff, and then they can do what?
Release it.
What's contractile mean? They contract. And they act sort of like, I don't have a pipette in here, but you know the little heads on the pipettes that you push in and it sprays the water out? That's sort of what a contractile vacuole acts like. It fills up with water, and then it contracts, and it pushes that water out of the cell. It's almost like a pump. Think of a contractile vacuole like a water pump. And for the amoeba, it always wants to pump the water which way? Out. Okay? So it's continually pumping the water out. If it ever stops, it's going to swell up and explode. Don't want that to happen if you're an amoeba. All right, one other thing I will mention, and then I'll stop, and we'll do a little stuff out of the book. something that we're going to do in the lab probably Monday or Tuesday. We're going to take some of that Elodea plant that we looked at, the plant out of the river. We're going to look at it under the scope again. And it sort of looked like this when we looked at it under the scope before, right? You had the nice little rectangles in there. And what do we say the rectangles were? Cell walls. It had all these little green dots in there. And my green doesn't show up on that green very well. What were the green dots? Chloroplasts. And they were kind of spread out all throughout the cell. The cell looked full. You might have seen almost like it looked like a little empty space in the middle. What was that? Looked kind of clear in the middle. What's the big thing in the middle of a plant cell? Big old vacuole. But the rest of the cell was full. What we're going to do Monday or Tuesday is we're going to take some really salty water. And instead of putting a drop of water on the plant leaves like we did before, the drop of pure water, we're going to put a drop of really salty water on it. Really, really salty water. We're going to let it sit there for a few minutes. After we do, those plant cells are going to look like this. Or maybe, might be easier to see like these. What's happened to those cells?
Dried them out.
First of all, what didn't change?
The wall.
The wall didn't change. This is hard, solid thing, right? It's not going to shrivel or do anything. It's going to stay the way it is. But what did shrivel? Everything inside what shriveled up? Everything inside the cell membrane shriveled. Because the water moved out of the cell toward what? Toward the high solute concentration. Water always moves from where there's less solutes, in this case inside the cell, to where there's more solutes outside the cell. One additional kind of thing. These are red blood cells, like the ones in your body. Over here, the red blood cells are in an isotonic solution. What's isotonic mean again? Same concentration of solutes as inside the cell. So if you notice, there's some arrows here showing you that water's moving in, but at the same time some water's moving in, there's also some water what? Moving out. It's even. Okay? There's no net osmosis. Is osmosis happening in this beaker? Yes. But the same amount moving in is moving out. There's no net osmosis. On the other hand, here we put the red blood cell in a hypertonic solution. What are all those little purple dots supposed to be? Those are solutes. And there's a lot of solutes outside the cell, but almost none inside, right? So which way does the water move? Out. And what's happening to the cell? It is shriveling and dying. Water moves from where there's less solute to where there's more solute. Over here, we put the cell in a hypotonic solution. And what's happening here, there's less solute outside the cell than there is inside. So water starts to move in. And that cell just starts to swell and swell and swell. And eventually, if it's an animal cell, it's just going to pop. Or if it's a plant cell, what's going to build up inside it? Because of the wall, what's going to build up inside that cell? A lot of pressure. A lot of water pressure is going to build up. One other thing to think about. Let's say you go swimming. And you're swimming in a freshwater swimming pool. And you're one of those people, when you swim underwater with your eyes open a lot. So you've been swimming underwater a lot for hours. You're out there all day long in the summer. You're swimming underwater most of that time, eyes open. You're maybe diving for pennies or something on the bottom. By the end of the day, what does your head feel like?
It hurts.
It feels a lot of pressure right in here, right? During the day, what's actually happened to your eyes? They've been sucking in water. Okay? Water's moved from where there's less solute outside in the pool to where there's more solute in your eyeballs. And your eyeballs literally swell a little bit. Not much, but a little. On the other hand, let's say you're one of those people who can open your eyes up and dive without a mask in the ocean. Okay?
Yeah, why does
I know people that do that all the time. Okay? Bothers my eyes. Well, I wear contacts. So I'm not gonna wear a mask anyway. But if you're diving in the ocean with your eyes open, what's going to happen if you do that for a long time? Your eyes are literally going to shrivel a little bit because there's less what in your eyeballs than there is in the ocean water. Less solutes. And water moves from where there's less solutes in your eye out to where there's more solutes in the ocean water. Your eyes shrivel a little bit, maybe a measurable amount. But that's osmosis, the diffusion of water. Let me stop the video there.